we are ready for session number 2 with the title Developments in Exposure of Humans to Micro and Nanoplastics. So here we have 4 talks, 2 of them are keynote, and the first keynote speaker in this session is Sabine Pahl from the University of Vienna, Austria. And the title is Human Exposure to Micro and Nanoplastics, What Drives Citizens' Concerns? So I give the word over to you.
Thank you very much for the introduction and thank you for the invitation today. This is very exciting. Maybe just a little explanation about my background. I'm a psychologist. I'm a behavioral scientist by training, and this is perhaps a little unusual in this context. But I have worked with Richard Thompson for many years, and I was also the vice chair of the SAPEA report that's already been mentioned. And on my slides, which I cannot see yet, I have the 2 affiliations, so I'm still doing work with Richard Thompson at the University of Plymouth, but I have moved to Vienna last year. So today I want to talk about citizen concern and attempt some explanation of the reasons, of the processes underlying this concern, which comes to a surprise to many scientists and also policymakers who are having to deal with this. And I will not distinguish too carefully between the macro, micro, and nano level because they are conflated in people's minds. I will refer to data using both, but it is difficult to really be very specific at this time. We need more data on this. So because of this, I want to start also by putting micro and nanoplastics and plastic pollution concern in general in context, building on Domagoy's great talk, Domagoy and Severina's great talk earlier. First question is, how concerned are people about plastic pollution in the context of other environmental issues? In this example, I refer to a study by colleagues in Australia. They had a representative Australian sample and asked a fairly straightforward question. Please indicate how serious do you think each of the following environmental issues are. The issues are on the right-hand side. They range from plastic in the ocean to other plastic sort of topics. And right at the bottom you see climate change, and I will show you the means and standard deviations here. The scale was from 1 to 10, so you can see overall concern is fairly high, but you can also see that the top rated items or issues are all to do with plastic waste and general waste. And that's about a point on the scale above climate change, which is quite interesting and has led to some discussions about the perhaps hype around plastic. To what extent are people over concerned, which I think is part of the discussion today. So this is before we even turn to human health. Let's have a look at human health next, but still stay in the context of marine threats, marine environments. So this is a study we did a couple of years ago using European plus data. It's actually over 15,000 people, 15 countries in Europe and Australia as well added. And what we asked people here is to tell us their concern, their worry about marine threats in terms of human health. So we specifically asked the question, to what extent are you worried about these threats because of human health? And there's a lot on this slide, but the ratings were from 0, not at all concerned, to 6, extremely concerned. And I've just highlighted on the slide the top 5 concerns. And you can see plastic pollution is right at the top, followed by chemical and oil pollution, then loss of marine species, contamination of seafood, and collapse of fish stocks. I think especially in the EFSA context, this is really interesting data, and it shows that people, when they think about human health, they make some connection to plastic pollution, but also to seafood, to fish stocks. The pattern is the same when you look at the countries individually. Apart from 2, Poland and Greece, you find plastic pollution and chemical oil pollution reversed. So I think this is interesting data because it makes the link between human health and marine threats. Now, we still don't quite know what people are thinking when they make this link. So the next study I want to refer to is a study we also did a few years ago, and this is a very small scale qualitative study, completely the opposite in a way of this large study. And it's the question was, what do people think about microbeads, microplastics in cosmetics? That's the question of the study. And I'm showing you data here because we had spontaneously people mentioning the link to human health, particularly to seafood. So I've highlighted this in the little red box at the bottom. Thoughts on human health. We had 3 different groups of people. Beauticians, that was relevant for the cosmetics. We also had students and environmentalists. And you can see the students are concerned about human health because of the microplastics in seafood. They say, oh yeah, this gets digested by animals, then you eat the animals, you're eating those, you can't afford to eat plastic. I mean, this is taken directly from their quotations. So they're just thinking it through. They're thinking about a mental model of how microplastics is worrying for them. The environmentalists, these were people who were more knowledgeable about the topic area. They talked about the food chain, a bit more technical. And this is also important, and it relates to Domagoy's and Severina's talk earlier, because these are mental models. These are things of how people think about issues and threats. And that's the study we're working on together. It's a mental model study that explores this link much more carefully and will really help us understand risk communications and risk perception in different audiences. So spontaneous mention, people do say this, even if you ask them about a completely different context. And then the final slide I wanted to show you, just to put the plastic concern in context, is actually the same data that Domagoy and Severin showed earlier. And I just wanted to say, compared to other food risks, we heard this earlier, microplastics are really, really high. What I find fascinating is actually that it almost looks to me as if the trend is going down slightly, and we need to work more with the BFR to find out if this is the case, because it's actually dropped compared to earlier data. So maybe we're already beyond peak microplastic concern. That's a hypothesis. We can discuss this further. So this gives you an overview. I hope it also shows how important it is to have good social data, to be able to follow these perceptions in the public over time. Because without this good social data, we can't really think about the policy and the policy impact in the general publication. So these are great resources. And the work by BFR in Germany and EFSA in this respect is invaluable. Also the Eurobarometer. So far we've looked, in line with other talks, we've looked at concern in the general public, representative samples, so this is pretty good data. But why is there this discrepancy to experts? Or do we actually know what experts think about and what experts think overall? We've heard Bart's talk earlier saying we're really lacking evidence. So next I want to go into the expert concern a bit more deeply. There isn't an awful lot of data. There's just been a new study, which I haven't been able to include here, but I give you 2 examples of data I've come across where we can infer some things about what experts are thinking. The first one is we took the opportunity to gather some evidence from microplastics experts around the Micro 2020 conference. It's a relatively small sample, but it's quite difficult to get experts to comment on these things. And we asked them to say how worried were they about macroplastics on the one side and microplastics, and in terms of the natural environment or human health. And what you can see here is one main effect, that people in this expert sample were more worried about the environment than about human health. In fact, we also find that in some public samples, so that's interesting. If you look at the human health, that's a sort of reddish bar. You can see that's middling. So the scale was 1, not worried at all, to 7, extremely worried. The human health, they're a little undecided. They're a little bit worried, but not extremely so. So that's one piece of evidence. There's also a new study that was mentioned earlier by Thiele and Hudson, where they talk about the uncertainties about risk associated with microplastics. It's a convenient sample. They go into specific health risks a bit more, and they talk about food, drinking water, and air. Those are the things that people mention. I didn't have time to include this here. The second thing I want to show you is from a piece of work we did for UNEP last year. And UNEP asked us to do a stock take of global actions to reduce the flow of marine plastic and microplastic to the ocean. This was a fairly big piece of work where we asked member states and major groups and other organizations who are active in this area to tell us about all the actions they do that target this. And actions here means 4 types of actions: legislative, standards and rules, working with people, so awareness raising, behavior change campaigns, technology and processes, and 4th, monitoring and analysis. It's a snapshot of action. It's not exhaustive. We couldn't achieve that, but that's the stock take. We had over 200 actions reported. You can see this here on the map. And one result I wanted to share, we did varied analyses on these, but one result I wanted to share was we asked people to report what type of impact their action was targeting. And I'll show you this. My clicker keeps going off. I'll show you this here. So what type of impact or harm does your action target? And you can see the top 3 mentions are marine organisms, so protecting marine organisms, protecting biodiversity, and the 3rd one mentioned is human health and well-being, followed by ecosystem services, and then the food chain. People were allowed to tick all of these boxes, so you have to take this with a pinch of salt. They didn't prioritize. They just said, what I'm doing, this big new law addresses these things. But what I find fascinating is that we've already heard earlier from Bart that the evidence for human health effects isn't really there at the moment. And yet people who are in charge of these programs, who are managing these programs, are telling us that they're partially doing it because of the human health impacts. So that's what I wanted to add here. So the picture so far is one where people are concerned, the general public is concerned, but there's a lack of evidence. And I think I have to speed up a little bit. I want to go into that topic of evidence and lack of evidence a little bit more. One thing to draw on is the SAPEA report itself, which Bart led. I was the vice chair on. And the main message that we published in the report that was taken forward and so on is the best available evidence suggests microplastics and nanoplastics do not pose a widespread risk to humans. And I just want to talk you through a few of the responses we had. And there was actually a lively discussion where we published further commentary papers by some colleagues of ours who said, where's the evidence that human exposure to microplastics is safe? So they were asking a question about the evidence that we presented. And we had to think, well, what are they asking us? Do they disagree? Did we not review the right sort of evidence? Did we not do it carefully enough? And so on. And we actually responded to this. And it was sort of going more towards a philosophical debate where we asked, what's the nature of evidence? If we keep looking for evidence, every single instance where we don't find it surely helps us feed into the policy process and so on. But what I find interesting is that there is clearly debate amongst scientists, and the scientists, even though it's supposed to be a neutral evidence-based process, bring in their own perceptions. So in this case, Leslie and DePledge were arguing we're not looking to the precautionary principle, whereas we looked at that quite carefully. So that's one instance. The other evidence about the lack of knowledge is from another study we did with an expert sample, but in the sense of an expert sample in industry. So these were waste managers and packaging experts and so on. We asked them what's the state of the evidence in terms of different aspects. And what I've circled here is the human physical health, the evidence from plastic pollution, and human mental health. And again, the judgment here is poor or very poor. Again, in contrast to that, there's been this paper in February 2021 by Vetak and Legler, where they say, yeah, we need to address knowledge gaps, but interestingly, they say to ascertain the health risks of microplastics. Again, I wonder if they're giving away a certain expectation or a certain perception that there is health risk and we just need to find it, which to me sounds like an interesting approach to scientific evidence. So on the whole, it seems there's a lot of debate. Scientists don't agree. The state of the evidence isn't great, and we're trying to make sense of this. So if we put this into the public domain, I want to give you some examples of what types of evidence communication people are receiving. There's an example, I hope, Bart, you don't mind, I've taken this from one of your talks, an example that shows a scientific piece of evidence. What it really is doesn't matter too much. It's a risk assessment about different species, but I just want to show you the really careful way this is presented. There's a threshold. The observed concentrations are related to this threshold. I hope I'm explaining this right, Bart. And this is distinguished for lots of different species. So this is a really complex piece of evidence analysis and a complex piece of representation. And in the SAPEA report itself, an additional effort the natural scientists went to really clearly show what is known, what is partially known, what is unknown. It's a very clear way of presenting uncertainty in a way. Now, if you contrast this to media messages, and we've seen some already earlier, that's nice to see in the other talks. Here's one from The Guardian, which is actually a really, I think, careful media outlet. And you can see it's an emotive topic, microplastics revealed in the placentas of unborn babies, but it says health impacts unknown. Scientists say particles may cause. Even comparing these two pieces of evidence, they show a very different type of communication. There's a picture there, quite an emotive picture, which I'll talk about a bit more, and they're clearly very, very similar. I've put some other newspaper headlines at the bottom, which I'll spare you the lurid detail. And one thing I wanted to point out is that when you compare different types of messages, you have different bases for this. You've got single studies, that's typically what you find in the media, versus synthesis, which is an approach that we take in science. You've got the presence of microplastics, for example, versus the impact of microplastics, and the two seem to be conflated. And I said this already, the reporting of uncertainty, of variation in the data, gets missed out in media reporting quite a lot. Now, as an even more extreme example, so I've shown you a science message, I've shown you a media message. What about NGO messages? They have perhaps specific interests, they want to activate people. And here's an example that I've come across that I think is fascinating because it's very extreme. It says plastic can cause birth defects, a very emotive picture again. And the text that goes with it is plastic can cause cancer, heart disease, and Alzheimer's. Chemicals in plastics can cause cancer. So they're changing their tack slightly. Scientific evidence is piling up. It's becoming impossible to ignore. That's just so different from what we see in the science domain. But this is what we have to contend with. And that, of course, explains partly the public concern. So let's turn to some of the principles that underlie these communications, the psychological ways in which we process this. I've given you an example here with a visual image for a reason, because the power of visual images is quite well researched. And I only want to talk briefly about the summary of neuroscience. I mean, visual processing is very dominant in people, in humans. 50% of the brain is used, that's an estimate. 70% of the sensory receptors are in the eye, so it's a very powerful way of communicating. And it's very, very quick, 100 milliseconds to get a sense of a visual scene. So this is where the careful science maybe falls back behind these emotive images. If you look at the psychology literature, we know visuals are important in terms of cognitive processes. They draw attention. You retain them in memory. They're quite easy to draw up again. There's a really fascinating area of research around traumatic images. Maybe this isn't quite a traumatic image unless you're a very passionate environmentalist, but they're really easy to remember and they flash back even if you don't want them to. So there are cognitive processes that are linked to this. There's a motivational element. You're interested in these images. They draw you in. You put effort in to think about them more carefully. You want to find more information. All good things if it's going in the right direction towards good evidence, and you want to elaborate them. There are emotional aspects. You might feel fear, disgust, anger. There are social aspects whereby you share them or you want to discuss them with friends and family. You really rely on them. And they may be linked to behavior, so they can provide cues for action. Again, if used in the right way, that's a really good thing. They can facilitate the breaking of habits. So I'm speaking as a behavioral scientist here. Beyond visuals, I think it's interesting to think a bit more about the impact and the spread. I mean, these are high-stake topics, high-stake messages. It's about human health. It's about baby health. It's unborn babies. I mean, what could be more topical? It's about male sexuality, microplastics in the placenta, so fertility. It's our food, the air we breathe. These are the messages we see out there. And I thought it's quite useful to refer to a piece of work done by not ourselves, other colleagues who looked at the spread of true and false news online. And if I summarize this, because I'm running out of time, they found that false stories were diffused further, faster, and deeper. In this instance, they were retweeted more, they were looked at by more people, and they were retweeted more. And I think one key aspect to take away from this study is that it was people responsible for this effect rather than bots, robots that retweet things. And it's people, it's human processes we need to look for. Right, I'm speeding up. In terms of general risk perception, there's a really great summary paper by Jenkins et al. That looks at risk perception applied to food, and she summarizes the factors that determine risk perception. And if we apply those to plastic, and I've put another example here a few years back in Canada, what we find is that people have higher risk perception for technical and natural hazards. That relates to Bart's slide earlier, where we compared particles from, I don't know why the slide keeps going forward because I'm not pressing the button, where he looked at particles made from different materials. And you might say, well, maybe if I breathe in a carbon particle, that's just as bad as a plastic particle. Technically, that might be true, but people are more worried about unnatural, about technical, about artificial things. And plastic is one of those. The risk target, we already said, vulnerable populations, babies, and so on. If it's about those, that increases risk perception. Number of people affected, we know plastic is everywhere. There's lack of controllability, there's delay, and there's the uncertainty issue. There's also correlation between risks and benefits. So if we think something has a benefit for us, we're more likely to accept it. It's interesting how that works out for plastic. We haven't got any data on this at the moment. I'm going to skip the example I was going to give about disgust just for time. And I just wanted to end on this social amplification of risk framework, which is an overview of all the processes that are happening. So from the sources of information on the left, which could be social media, to the information channels, how do you come across this, to social stations. This includes opinion leaders who might give you messages about plastic. I can think of David Attenborough as a very powerful opinion leader who has been talking about plastic a lot. To individual stations, this is where the psychological processes come into play. They then have effects on institutional and social behavior, including social protest. That's the right-hand side of the rectangle. And then if we skip to the very right-hand side, the impacts are varied. They can be financial losses, loss of sales. They can be an increase or decrease in physical risk because people change their behavior or because things are regulated. And they can also include loss of confidence in institutions. If an NGO keeps giving alarmist messages, they might well lose trust in the audience. So, final slide. Apologies, I'm slightly over time. A few key messages. There's high level of citizen concern about macro and microplastics, and this includes human health impact. The evidence is now emerging. Experts are more concerned about environmental impact. In fact, that's also true of non-experts, but there is a lack of scientific evidence and there's great debate even among the scientists. People are exposed to different media messages, including those visuals, and we can look to psychological and social processes to explain these responses. And in some instances, we can explain why social amplification follows from this exposure. And the final 2 points, I think, to me, are perhaps the most important. Risk is a societal issue between the technical risk assessment. So some people are really keen on that. But there's also the values, emotions, the trust. And they're not necessarily one is right or wrong. We need to bring the two together beyond the mere facts. And we need to understand public concern and behavior to ensure policies are successful. And I'm thanking all the people I've worked with for a long time. Thank you.
Thanks a lot for this interesting talk. I'm afraid I had to disappoint you with the questions, because we have questions, but there's no time for them.
Sorry.
So I'm sure we can return to the discussion tomorrow. So I think that's better than, in order to respect the time of the rest of the
Sure.
speakers. So we move on to the second keynote lecture, and that's given by Francesco Coppada from the Italian National Health Institute, and the title is Exposure to Micro and Nanoplastics via Food and Feed. So, Francesco, you have the floor.
Thank you. Thank you for the introduction and for the invitation. By the way, as just said, my name is Francesco Coppada, and I work at the Italian National Institute of Health. A few words about myself. My activity and research interests include, on the one hand, exposure assessment of chemicals, and on the other hand, particle toxicology, analytical determination, and risk assessment. And I've been working as an expert for several EFSA working groups in the last 12 years, including the one that dealt with the micro and nanoplastic some years ago, and the present ones dealing with nano-related issues. So my presentation is divided into two main parts. The first is the microplastic contamination of selected food chains and what the resulting human exposure might be. And I will briefly touch upon the marine environment, terrestrial environment, food processing as a potential source of microplastic contamination, and drinking water. Then I will expand on the knowledge gaps preventing dietary exposure assessment, and touch upon potential way forwards, focusing especially on particle physicochemical properties, on the issue of size range from a risk assessment perspective, and on the limitation of the existing data and how we can go beyond these limitations in order to achieve exposure assessment. So the presentation relies on some important scientific opinions and reports which appeared in the last years, some selected reviews which appeared in the last 2 years, and some dozens of individual peer-reviewed publications which are relevant for the topic. This morning we have seen that a significant proportion of the plastic that is produced and used is not disposed of properly. It is dispersed in the environment, it may persist in the environment, and this is true especially for the marine environment. So there is a lot of focus on the marine environment and the impact of on seafood. Plastic litter comes with many different types of plastics involved, but polyethylene, polypropylene, and polystyrene are the main 3 polymers involved. This marine litter may fragmentate and degrade to the so-called secondary microplastics, which are indeed the main source of microplastic in this environment, whereas primary microplastics likely play a minor role. Further degradation of these plastic particles may lead to nanoplastics. Microplastics can be ingested by both marine invertebrates and vertebrates, and they are exposed either directly or via lower trophic levels. So, as most of the larger size microplastic remain confined within the GI tract of fish, it has been put forward that gutting is expected to decrease human exposure. This, however, does not apply to shellfish and does not apply to certain types of small fish, and it may be less applicable also to smaller size particles. Actually, there are papers showing a correlation between particles in the GI tract of fish and particles in the muscle tissue. It is clear that filter feeders such as mussels do represent the worst case for plastic particle bioaccumulation, and their consumption may lead to high exposure levels. Next slide, please. For terrestrial food chains, very little is known. Microplastic may enter agricultural soils through the use of savage sludge as a fertilizer, may also come from degradation of plastic materials used in agricultural practices, such as plastic mulching. And indeed, it is been shown that fish meal is used in some animal production areas, and microplastics from marine sources might actually end up in non-marine foods. Globally, there is a huge knowledge gap about the potential uptake and deposition of plastic particles in vegetable and animal food. Existing data have limitation. There are data on matrices like sugar, salt, or honey, which partially indicate that aerial deposition might play a role, but overall there are huge limitations about other and more important matrices. Next slide, please. So food processing is another potential source of contamination. It may include several different sources of microplastic in human food. It is plausible that certain processes release plastics, and aerial deposition, for instance, of fibers, might also play a role. We know that plastic is used in a number of food and beverage packaging, and the actual release of plastic particles might change with processing conditions and might increase upon recycling or reuse. Drinking water is clearly an item of importance in terms of potential human exposure. Occurrence levels seem to be higher in bottled water. Lower contamination levels have been shown in water sold in glass bottles, and lower levels appear to be present in tap water. In general, freshwater ecosystems are less studied compared to marine ecosystems, but indeed surface waters, which can be used as a water supply, might be heavily impacted, whereas groundwater is generally less impacted. And this morning we have been told that by the end of 2024, microplastics might enter the watch list of the EU Drinking Water Directive. So targeting relevant size ranges will be essential in this respect. Indeed, next slide, please. Indeed, physicochemical properties are key for the assessment of micro and nanoplastics. Experience with particle and fiber toxicology does indicate that any potential adverse effect of these particles will be dictated by properties like size, morphology, surface properties including surface charge, and the chemical composition of the particles, that is, their constituting polymers. And the chemicals contained in the particles or absorbed onto the particles might be other determinants of toxicity. Another key point that has been already touched upon this morning is that smaller particles do come from fragmentation of larger particles. So differently from other types of particulate contaminants, environmental plastic particles cover a broad size range extending over 6 orders of magnitude. And this is very peculiar to micro and nanoplastics, because normally other particulate contaminants come with a much narrower size range. In the figure, you see the size classes found of microplastics found in oysters, and you can see that the smaller the particles, the higher the number in the tissues. And this is a general law, and power law size distribution has been proposed to also predict from larger particles the amount and presence of smaller particles in quantitative terms. Next slide, please. So from this point of view, this broad definition of microplastics, which encompasses a wide particle size range, is a problem when it comes to estimation of data exposure. This is a table taken from a recent review where estimated exposure to microplastics from the diet, from drinking water, and from excretion from human stool have been estimated, and you see on the right these estimates expressed as number of particles per kilogram body weight per day. And when the whole diet is considered, the amounts are in the range of hundreds of particles, but then there is a variability which is quite marked. But what is really important here is that if the particle size is considered, these individual studies span from particles sized 1 micrometer to 500 micrometers. And such studies focusing on food matrices, which investigate different portions of the size continuum over which microplastics occurs, are really comparing types of particles which cannot be compared. For instance, if we take a spherical plastic particle with the same composition and with 1 micrometer in diameter or 50 micrometers in diameter, we see that the larger particle has a mass equal to more than 100,000 of the smaller particles. So exposures expressed in terms of number of ingested particles of different size are really not comparable. They are intrinsically different entities, and translation of such exposures on a mass basis, for instance as microgram per kilogram body weight, would be even more misleading. So this is a key point that has to be kept in mind. It is also to be considered that environmental plastic particles are irregular in shape. Fibers predominate in certain size ranges, and this calls for additional caution when particle size is dealt with. So expressing exposure in terms of number of particles is to be very, very carefully considered. But the most important point is that none of the investigated particle size ranges is related to established human health effects. Next slide, please. So this is an important aspect, but there are other limitations of existing data that prevent a direct exposure assessment to be carried out. In general, the plastic particles which are likely to be relevant for human health impact are limited to the smaller size microplastics and the nanoplastics, but there are no methods for this size range in terms of particle measurement. Other shortcomings are the lack of harmonization in analytical techniques, the limited availability of particle composition data, and the poor, in many cases absent, analytical quality control. All these aspects have been highlighted this morning. Analytical quality control is essential for analytes that are in most cases ubiquitous. Next slide, please. So the issue of the size range is indeed a key one. A univocal regulatory definition is often invoked as the necessary prerequisite for harmonization of methodological approaches and comparability of data. However, whereas this definition is essential from a regulatory point of view, in terms of human health risk, toxicokinetic consideration should be the starting point for prioritizing size ranges for hazard and exposure assessment. For the nanoplastic, a reference is the size range considered for nanospecific risk assessment by the relevant EFSA guidance, and this size range goes up to 250 nanometers. Because there is evidence of intestinal cell uptake of particles via endocytosis and other pathways within this size range. On the other hand, for the microplastics, there is some evidence that translocation across the mammalian gut may happen for particles up to a few of micrometers. And the EFSA 2016 statement highlighted that only the smallest fraction size less than 1.5 micrometer might penetrate deeply into organs. So, based on the different routes for potential crossing of the intestinal epithelium and the likely different fate in the human body, relevant size ranges should be established in order to prioritize hazard and exposure assessment. So for nanoplastics, size range up to 250 nanometer appears relevant, whereas for microplastic, a size range from 250 nanometers to approximately 1 micrometer or few micrometers appears to be relevant. And this is clearly an area of important research and development. Next slide, please. So it is clear from the previous discussion that any development in exposure assessment of plastic particles relies on both progresses in toxicology and in analytical methods. Research on plastic particle toxicokinetics and toxicology is indeed key, and this especially is true for the toxicokinetics of plastic particles, which is still largely unknown, and also for the toxicology of these particles in relation to their physical-chemical properties, focusing on effects of long-term exposure, keeping in mind that dietary exposure is lifetime exposure. The effect of aging is also relevant because test particles might be not entirely representative of environmental plastic particles. And also another topic of interest is if contaminants absorbed onto particles might have their toxicokinetic behavior changed by the particles. So if the particles after absorption might be carriers of these contaminants within the human body, or even within cells, which would be a sort of potential Trojan horse effect. And analytical methods, sorry for this. Another key point is development of analytical methods, and indeed these methods should be capable to measure relevant particles that are to be prioritized based on hazard assessment, at the expected low background levels in food. So this is another key area of development. So summarizing with a few remarks, this discussion, first point is that clearly we deal with a problem of comparability of data. This is due to the fact that microplastics are studied with approaches which are often metric specific. This is inherent to an evolving field of research, but it indeed poses a challenge for exposure and risk assessment. And the second point is that occurrence data of acceptable quality are very limited, and there is a paucity of chemically specific data, that is, data related to the particle composition. And a further limitation is the lack or paucity of analytical quality assurance and control in the existing studies. After oral exposure, we have seen that size morphology, surface properties, and chemical composition of the particles will determine their ability to be taken up in the gut and cause any systemic toxicity. So this is a key aspect for further research. And hazard assessment is really key in order to identify relevant size ranges to focus on, and the best metrics for exposure assessment, which may be number-based concentrations or mass-based concentrations, and any potential conversion approach. As far as nanoplastics are concerned, occurrence and exposure data are lacking. The need for analytical method development and exposure assessment should rely on solid evidence from hazard assessment. And development of analytical methods capable to determine plastic particles at the low background levels in food is another final important point. For the smallest nanoplastics, it is really key to prioritize the development of methods only after solid evidence from hazard assessment pointed out the need to do so, because developing this method will certainly entail a considerable technical challenge. Thank you for your attention.
Thanks a lot, Francesco. This was very interesting and touching into hazard in addition to exposure. So it's a list of questions for you, and we have some minutes. So the first one is from Liz Colebrook, and the question is: has anyone looked at the contribution of water used in food processing to micro and nanoplastics content of processed foods?
Yes, there are indeed some studies pointing at the fact that the water component during food processing might actually contribute to the final concentration levels in the processed items. Of course, as I said, these studies are on their own respect interesting, but have limitations because all of them focus on specific size ranges. So it is really difficult to say if this is a general aspect or it is not. In general, we can expect that tap water is present lower contamination levels, and sometimes also it has to be taken into account that the water used in food processing is further filtered and processed. So this is certainly something that has to be studied more in detail.
Mm-hmm. Thanks. And the next question is from Susanne Beltz. The question is: what in your opinion, is the best parameter for regulatory purposes and risk assessment? Number or mass or something else?
Well, this is not an easy question. In general, I think that this is still a very debated issue also for nanomaterials in general and for in the area of nanotoxicology, where also other properties like surface area might be relevant for certain type of exposures. But what is most important is to actually realize that these particles looks very differently, and we have to talk with great caution about size, because size is the size of particles spanning a wide range of different morphology, from fibers to irregular polyhedric particles, and in principle, it is possible to convert larger particles to smaller particles, and there are some attempts also to infer about this transformation of number of particles into mass of particles using, for instance, the ellipsoidal shape, but all this modeling has to be considered as an interesting approach, but indeed with many uncertainties.
So not easy to just pick a choice.
Yeah.
So the next question, we have still some time, is from Henriqueta Loro. Thank you very much for this interesting talk. Human internal exposure was shown in human colon specimens, and reference to Ibrahim 2021. Thus, human intestine accumulation is of utmost concern. What about the leaching of the plasticizers in the organism, such as phthalates or new generation plasticizers, stinge? Is there any known risk?
This is a very interesting question indeed. Normally, the aspect of chemicals carried by plastics is seen from the perspective of potential release in the GE tract. But indeed, there might be that absorption of plastic particles is a way to have these chemicals released in the human body, both in body fluids, but in principle also, as I said, within cells for the smaller particles, for the nanoplastics, for instance, and this would resemble a sort of Trojan horse effect. So in principle, this effect might happen, but there is no clear evidence about them, and this is another area that have to be studied more in depth.
Yes, thank you. And it will also be touched upon in later talks, the contaminants in the plastic. So I don't think we have time for more questions, and we can move over to the next talk. And that is, the title of that talk is Assessment of Interactions of Nanoplastic and Wheat Using PD-doped Plastics. First Insights on Potential Uptake and Impact. And this talk is given by Ana Elena Pradas de Real from the University of Madrid. So please, you have the floor.
Good afternoon, everybody, and thank you very much for the presentation. So today I'm going to present a project that has been performed in collaboration with my institute, l'Institut des Sciences de la Terre de Grenoble in France, the European Radiation Facility also in France, ETH Zurich, and Exploration from Berlin. So in fact, there are some scientists that, based on models, say that soils may be one of the largest plastic sinks in the environment, and that even we can have more plastics in soil than in the oceans. And there are two main sources of plastics arriving to soils. So in one hand, we have the application of sewage sludge as fertilized. So the plastic that we use in our daily life, over their life cycle or at the end of the life, they arrive to wastewater. And in wastewater treatment plants, they are retained on the sewage sludge. The sewage sludge is quite rich in organic matter and in nutrients, so it's used as fertilizer in agricultural soils. So it is estimated that in between 63 to 430 kilotons of plastics arrive to European soils through this pathway. And we have already some studies that have identified some polymer fragments and fibers in this sewage sludge. But then we also have the extensive use of plastics in agriculture, mainly for mulching, but also for the building of greenhouses. So from these two sources, when the plastic fragments arrive to soil, due to solar radiation or for the mechanical action of plant roots and vertebrates and invertebrates in soil, they break down to lead the microplastics and nanoplastics. So this micro and nanoplastic can be uptaken by soil organisms and by crop plants. So we wonder whether we can have a transfer to the food chain if we consume these crops, and also whether the presence of this plastic litter in soil can compromise the ability of agroecosystems to provide food. So today I will focus on, in the case of nanoplastics and the interaction with crop plants. The problem is that we have already mentioned today is that the information in this case is also very limited. We have very little information about nanoplastics and plant interaction. And of course, one of the main reasons is the lack of detection technique. Here I show the example of polyestyrene and humic acids and how difficult it can be to distinguish this. For the moment, we only have 3 studies reporting concentration of plastics in soil. And these concentrations go from 50 to 500 milligrams per kilo. But we have no information about nanoplastics. So these concentrations, due to the sampling and analytical methods, are mainly focused on the micro and macro sizes. However, to overcome this lack of knowledge, some studies have used fluorescent nanoparticles as an alternative. And these studies have bring interesting knowledge. For example, we know that nanoplastic can be accumulated on the coat of seed, interfering the seed germination. We also know that some nanoplastics can be uptaken by plant roots, and we even have an example of how nanoplastics in soil can be transferred to plant and then can be transferred to, in this case, snails. So we already have an example of transfer to the food chains. The problem is that the use of this fluorescent nanoplastic presents some limitations. The first one is that they are limited to the direct observation under a microscope. Then we can also have some artifacts such as the fluorophore leaching and the tissue autofluorescence. And the other problem is that they do not provide quantitative information about the uptake and the distribution once in plant or animal tissues. And then also in these studies, we found contradicting results regarding the phytotoxicity. And this is related with this limitation and the fact that with the available information, we are still not able to establish the mechanisms that allow us to uptake, transport, and toxicity. So this status quo does not allow us to establish the risk associated with the presence of plastic litter in soil and its transfer to the food chain. However, even if the information that we have regarding nanoplastics in agroecosystem and nanoplastics interaction with plant is quite limited, we have an extensive knowledge about the fate and effect of engineering nanomaterials in the same agroecosystem. So the last years working with these engineering nanomaterials, we have learned that we need to work at realistic doses, the importance of particle parameters that have been already mentioned, like site, safe, and sulfate shares. For that, we will need a methodological development. We know that we need to consider the aging process in the environment, and in this case, the aging of particles in soils. One example is the formation of ecocoronas. And we have seen a lack of monotonicity in dose response. So here I'm showing an example of the nitrifications in soils exposed to different concentration of titanium oxide nanoparticles, and we see how this response it doesn't follow a classical dose response curve. So that means that we have to focus in mechanisms more than in the effects in order to understand what is going on. So in order to be able to perform these mechanistic studies, when we still don't have the methodological development necessary for that. What we propose is to use palladium-doped nanoplastics. So in this case, we have used polyester nanoplastics with a palladium core so that we can analyze the palladium as a proxy to detect the nanoplastics. That allows us to provide quantitative information, to use common equipment, and to perform mechanistic studies. But this is also very useful to validate some new methods that we can use for the real nanoplastics. So like this, we have exposed wheat plants in hydroponics to two morphologies of these nanoplastics, smooth and raspberry, and to two concentrations, 3 and 30 ppm, over 3 weeks. So our first question was to know whether the nanoplastics are absorbed by wheat plants. And for that, we used the palladium signal to determine nanoplastics concentration by using ICP-MS. So what we found is that wheat absorb nanoplastics, and then once in the root, these nanoplastics are translocated to the shoot. And it was in a dose-dependent manner. However, we didn't find any difference between the two morphologies tested in this study. So then our next question was to know, once we know that nanoplastics are absorbed, by which structure are they absorbed in plant? So for that, we first perform a scanning electron microscopy in root prepared by critical point drying. And here we can see the surface of a control root and the surface of a root exposed to 30 ppm raspberry nanoplastics. And we can see how the nanoplastics are agglomerated in the surface of the root and strongly associated with filaments that may correspond to the root MUC11. Then, in order to further investigate this uptake route, we took advantage in this case also of the palladium core, but to use the fluorescence of palladium. So we perform synchrotron micro XRF fluorescence in root teeth. So here we can see a control root, and we see the distribution of potassium in yellow and phosphorus in blue. And then we have the root of a plant exposed to 30 ppm raspberry nanoplastics, and beside the potassium and phosphorus signal, we also see the palladium signal in pink, showing the distribution of nanoplastics, mainly in the epidermis and in the root cap. So that indicates that the root cap is a main point of entry of nanoplastics. This has already been shown for engineering nanomaterials and for other pollutants, and this is because here the tissue, the cells, are quite disordered, so there is more space for the nanomaterials or nanoplastics in this case to be absorbed. So following the same approach, we also performed XRF fluorescence, but in this case in root cross-section. So here we have a cross-section of the root, where we can see the central cylinder, the cortex, and the epidermis. So we have the control root up and down the root exposed to the 30 ppm raspberry nanoplastic. So as I mentioned before, in this case, we are using the fluorescent signal of palladium. And we see in pink how the nanoplastics are accumulated in the epidermis. And by using higher resolution XRF, we see how these nanoplastics are strongly associated with the cell wall. So once we know that the nanoplastics are absorbed by plants and by which structure, we wanted to know if we have some effect on the plant. So for that, we used infrared spectroscopy, not to localize the nanoplastic, but to study the biochemical alteration in roots. So here I'm showing the principal component analysis and linear discriminant analysis of the infrared spectra collected from the exposed root, and we see a clear clustering, meaning that exposed root shows biochemical alterations due to the exposure. So in order to know what were the infrared bands related with this difference, we performed logistic regression with lasso estimator, so an statistical analysis to identify the main infrared bands explaining the difference. And in summary, what we found was an increase in all the bands related with cell walls, meaning pectin, lignin, cellulose, and carbohydrate in general, which point out to a reinforcement of the cell wall. So to confirm this, we used synchrotron microcomputed tomography. So with this technique, we can have a 3D reconstruction of our root, and we can separate the cell volumes from the cell wall. Then once we have the cell walls, we were able to perform a threshold based on cell wall density, and we classified this threshold in high, medium, and low. So here we have virtual cross-sections, slides of the roots, and we see how, especially in the roots exposed to a smooth nanoplastic, we have an increase in the high density region. This is more or less the same that we have in the graphics when we see the proportion of voxels. So this confirms a reinforcement of the epidermal cell wall, which is a defense mechanism observed in plants already for other kinds of pollutants, but never before for micro or nanoplastics. So our next question was to know if this change in cell wall density have an effect on root morphology. And for that, we used the same approach, but in this case, we investigated the cell volumes in the epidermis, in the stem, and in the vascular cylinder. And we found that the epidermis of exposed cells shows an strongly decreased cell volume. So in summary, what we have found in this study is that nanoplastics are absorbed by wheat and translocated to the shoot, that the root cap and the maturation zone of roots are main points of accumulation and absorption. We have observed a reinforcement of the root cell wall and change in root anatomy. And we have demonstrated that the use of palladium-doped nanoplastics allows to perform mechanistic studies while techniques to detect the nanoplastics are still under development. So we can use the same approach to go some step forward. For example, we can use this approach to investigate other uptake routes, like root hairs or cracks in the root, to investigate the transport route, so how the nanoplastics are transported what in the plant. And for that, we can also think in increasing the spatial resolution of the used technique. And we will need to move also to a more realistic experimental system in soils and in long-term experiment. For example, to investigate the consequence for plant development and for the nanoplastic fate. And one of the main findings of this study is that nanoplastic can be transported from root to the shoot. But in order to establish the real risk for transfer to the food chain, in the case of wheat, we will need also to investigate if the nanoplastic can also be transported from the shoot to the grain. And for that, we will also need to improve the spatial resolution and the detection limit of the technique that we have already used in this study. So I want to acknowledge all the team that have participated in this project, and I want to thank you also for your attention.
Thanks a lot for another interesting talk. There are no specific questions from the audience, but there is one from me. And so far, seafood has got most of the attention when it's come to exposure. But based on your talk, perhaps the terrestrial food chain or the plants is more or less overlooked as a source. You have probably made yourself some thoughts on that, so perhaps you could elaborate a little bit on that.
So sorry, I didn't hear the last part.
Sorry, I was thinking about whether you thought around this, whether plants could be perhaps as important or even more important than seafood when it comes to exposure. So far, the seafood has gained most of the interest. So would you like to comment on that? What do you think?
Yeah, I think that in fact we have For example, for engineering nanomaterials, we have seen that the risk will be quite low, but it seems that in the case of nanoplastics or plastics in general, we may have extremely high doses of plastics in soil. And in this study, we are using wheat, and the main source of consuming wheat will be the grain. But when we were doing also this experiment, we were thinking of the fact that we consume some crops from which we directly eat the leaves. So meaning that the travel of nanoplastics or microplastic from the soil to this edible part is much shorter. Or even if we think in other plants such as carrot or onions, that seems quite direct. And of course, we need to do much more studies, but it seems to be already to have some evidence that the plants are able to absorb nanoplastics. And even there's already one study showing that they can even absorb microplastics. So the question will be more that if the concentration are, big enough to be a risk for humans or not. But that we can find that the plants are able to absorb, for me, it's quite sure.
So thank you. It was a hard question. So I think it's time to move on. Thanks a lot. We have one more talk in this session, and that is from Jana Weiser at the Technical University of Munich. And the title of her talk is Entry of Microplastics into Packed Food and Beverages, the example of bottled mineral water. So please.
All right, thank you for the introduction. My name is Jana Weiser. I am a PhD candidate at Technical University of Munich, more exactly in the group of water systems technology, which belongs to the chair of food chemistry and molecular sensory science, even though I am not a food chemist myself. But I got involved in this project called MIPAC, which stands for Microplastics in the Aquatic Environment and in Foodstuffs, which is quite a big project. We are 5 chairs of Technical University working together with in total 15 companies coming from the food producing sector, food packaging, water treatment, and analytics. And the project is funded by Bayerische Forschungsstiftung. And in case you want to find more about it, because there's quite a lot of fields that we work through, you can check out our website if you like to. So today I want to talk about entry paths for microplastics into bottled water. All the results that I'm presenting to you today have recently been published in the journal Water if you want to have a look at the details. So I believe that if one day we want to probably minimize microplastics in food and beverages, we should find out about how the particles get into there. And in the case of mineral water, where we all know that microplastics are present depending on the bottle type, speaking of PET, glass bottles, single-use and reusable bottles, we all know that microplastics are there, but we don't know exactly where they are coming from. And the presumption that was made earlier by colleagues that in the case of reusable bottles, the bottle cleaning process is the critical point for microplastics to enter. So for this reason, we chose to focus our study on still mineral water and reusable glass bottles. And I'm very grateful that our project partners for mineral water bottlers gave us access to their bottling process, which I want to show you very quickly in this scheme that we have here. So in general, the process is the same everywhere. Mineral water is sourced from a well from the ground. And then in Germany, we have a lot of ferric ions in the water in many places. So they are removed through aeration and filtration. And we took another sample from this deferrized groundwater. The glass bottles that are coming back from the customers are cleaned in caustic baths. In some cases, it's only one. In some others, it's more caustics, but it's always soda with some tensates in it. And then the bottles are jetted with fresh water, and we also took samples of these cleaned bottles. Then the water is filled into the bottles, and bottles get their caps on. In our case, with the glass bottles, it was aluminum caps with a polymeric sealing layer inside. And of course, we sampled the final product as well. For all these different kinds of samples, we had to pursue different ways of how to prepare these samples for analysis. So in the case of raw and deferrized water, we took samples of at least 1,000 liter by filtering in situ through stainless steel cartridge filters. And in the picture in the bottom, you can see how bad it can be, even though it's only water, because of these ferric ions they precipitate. And we got rid of them through a density separation in zinc chloride solution and a citric acid cleaning. Citric acid treatment. The bottles made it a little easier for us. We took 3 bottles, flushed the water out, and then rinsed the bottles twice with a sodium dodecyl sulfate solution and once with ethanol. All the samples were deposited on anodisc filters for measurement later on. The caustic cleaning solution from the bottle washer was the toughest, I would say. Here I brought a picture for you to see how it can look like. There's quite a lot of solid substances in that. So we applied a Fenton's reagent together with ascorbic acid and a cellulose treatment to get rid of the solids, but in some cases we were still only able to analyze a subsample of the 1 liter that we took from the bottle washer. So as I said, we had the particles of the preparation on a anodisc filter, and we imaged that one with an Agilent Carry 620-670 FTIR imaging system equipped with a large FPA detector. And we used a 15x objective, which resulted in a 5.5 micrometer projected pixel size. So that means for every square of 5.5 by 5.5 micrometers on the sample, we would get 1 spectrum that has to be interpreted later on. With this technique, we could successfully analyze particles down to a size of 2 pixels, say 11 micrometers. And we imaged around a third of each filter, which resulted in 2 million spectra. So quite a lot of data to be analyzed. And we decided to do that using a machine learning approach, namely the random decision forest model, which we programmed in the software Epina Image Lab. Just very shortly to give you an idea what that means. We sampled around 6,000 example spectra of the most relevant polymers. Additionally, we included PLA as an example of a biodegradable polymer and cellulose and proteins as typical sample contaminants. The program was further provided by us with spectral descriptors, as are seen in the bottom picture here. Spectral descriptor is, for example, to tell the program to look at if there is a band at a certain point or if the band has a certain pattern and so on. We validated the program using a Monte Carlo cross-validation, which resulted in an overall model accuracy of 95.45%. So most of the assignments that the model made were correct, but still we went through all the results manually and checked and corrected where this was necessary. During all stages of sampling, sample preparation, and sample evaluation, we followed strict quality assurance and quality control measures. I don't want to speak too much about this right now because you can actually find all the details in a poster of this event. So I would really recommend to take a look, and soon it will be available as a paper as well. What I want to highlight here is the lessons learned from recovery rates and blank samples, which is that the more complicated a sample preparation process is, the more risk you have to lose particles on one side, so to have a low recovery rate. And then on the other hand, you have a higher risk of contamination from outside, which then results in higher LOD values, limit of detection values for this kind of sample. So we're getting to the results finally. We will start with the raw and deferrized water. So in raw water, we detected between 48 and 170 particles per cubic meter. Most of them were made up of PVC or polyesters, and as is typical in microplastic research, the smaller particles in the range of 11 to 50 micrometers were the most abundant. Most particles were fragments in our case, and as you can see in the scheme, there is a filter installed, and after this filter, as you could expect, the particle concentrations were very much lower, with only up to 53 particles per cubic meter. So this is obviously the filters do what they're supposed to do, and the numbers that we found are around the range that has been reported in literature in studies that applied similar methods. So in total, when you break it down to 1 liter of mineral water, the water itself is nearly free of microplastics. Then we got to the bottle washer, where, as you may remember, we thought that it would be the most critical point of entry for microplastics into the bottles. And indeed, the caustic cleaning solution was full of microplastics with up to 3,240 particles per liter. Most of them were polyethylene, and one sample we had quite a high share of polypropylene. So we were excited to look at the bottles that were cleaned with exactly this liquid, and to our surprise, all these bottles were below limit of detection, so they were nearly free of microplastics. But what I have to stress here is that, as I told you before, we were only able to analyze particles down to a size of 11 micrometers. And FTIR spectroscopy is not capable of detecting every substance that is probably there in these cleaning solutions, because there's also pigments from the bottle labels, and there is an Raman spectroscopy, for example, is able to detect these substances. And in fact, there is a study that did find microplastics and pigmented particles in the size range below 11 micrometers in cleaned glass bottles. So it looks like we have to distinguish here which particle size range we are talking about when speaking of this bottle cleaning process. So the clean bottles were like free of microplastics, and then we sampled bottles where the deferrized mineral water was filled in, and again these bottles were below limit of detection. But then when the bottles got their caps on, we saw a very sharp rise in microplastics concentrations, up to 75 to 700 particles per liter. Most of them were polyethylene and polystyrene, and we had again the smaller size fractions dominating here. Now, particle numbers and composition are very much related to what was known already in literature, but this especially high share of polyethylene, we wanted to dig deeper into that. And as is known from a study from Italy, from Anna Winkler, the capping, the twisting of bottle caps can release particles. So in their case, they took PET bottles with PE caps and twisted them several times and saw that there is abrasion at the bottle caps and the necks. So we took a look at this polymeric sealing layer that was in the aluminum caps our bottles were capped with. And here you can see at the top a spectrum of one of the PE particles that were detected in bottled water. And at the bottom you can see a polyethylene reference spectrum. And I'm sure you can spot a difference because there's another band there in the particles that is not there in polyethylene, even though it's still a very, very similar spectrum. And so we took a spectrum of a cap sealing layer, and in fact it's the same substance from what we can tell from the FDIR spectrum. So we have the same case here as the Italian colleagues had, that there's abrasion from the bottle cap. Of course, we wanted to know more about what material this is, and unfortunately the manufacturers are not so open with that, so we only could find out that this was a PVC-free soft polyolefin, which is available on the market in a broad range of qualities. And remarkable, the two samples that we had with the highest microplastics concentration had caps that used the same sealing variants. So I think that maybe these variants have different resistance to abrasion. To put it all into a nutshell, not our recent study, but what we know in total about microplastics and bottled water, is that the bottle material and the type, so PET, glass, reusable, single-use bottles, all play a role for entry paths into the bottled water. Cap abrasion, as we just saw, is the main entry path. Other studies suggest that carbonization and bottle age do play a role as well. We could show that the filling process itself, so everything that comes before capping the bottle, did not play a significant role. However, when it comes to the bottle cleaning process, especially particles below 11 micrometers may not be completely flushed out of the bottles. To give some implications for the future, I think that it's worth to take a closer look at these cap sealing materials, and also this may be true for any other kind of food packaging. There are ways to remove particles from the bottle caps, because we also found other substances, not only the bottle cap sealing. So one could try suction or rinsing devices here. It is still a good idea to keep bottle washing liquids clean, because everything that's not in there can never get into the bottle. I would not suggest to further reduce the water amount used for bottle jetting, even though from a water saving point of view, this is nice, but when you want to avoid microplastics, you should not reduce that. And I think that when it comes to reducing microplastics in food, science and the food industry should work together, because this way we can gain insights that otherwise we could never ever have. So I want to thank all my colleagues who worked with me on that. And I also wish to thank for the opportunity to speak here. Thank you very much for your attention.
Thank you very much, Jana, for an interesting talk. It's good to hear that you are able to identify where it's coming from. There's one question from the chat, and that comes from Tobin Robinson, and it says, does this imply that the reuse of, example, water cooler plastic bottles needs to be rethought?
Probably yes. So well, we only looked at glass bottles, but there's another study on reusable PET bottles, and unfortunately, they had the largest numbers of microplastics, even though reusable PET bottles are very favorable from an environmental impact point of view. If you want to avoid ingesting microplastics, then I would not recommend to drink from reusable PET bottles, unfortunately.
Thank you. I think there are no more questions, and it's time for a break. The break is planned to be lasting for 20 minutes, and I remind you to have a look at the posters if you have time for that. And we will start again at 15:50.